Yes, thank you for the introduction, and I'm very grateful for the invitation to be here. And I'll talk about the development of production method for a bioactive. Mar gave a very detailed introduction to chitin and chitosan in his talk, so I will not go into detail, but as it has been mentioned before, chitosan is derived from chitin, which is very abundant in nature and is mostly in crustaceans, arthropods, and fungi. And chitosan is obtained with chemical deacetylation of chitin and has significant antimicrobial activity. And it is a material that is non-toxic, biodegradable, and biocompatible. So it makes it possible for very versatile applications in the medical, nutraceutical, cosmetic, and food agriculture fields. Just to mention a few examples, it is used in wound treatment and bandages. It can be used as environmentally friendly packaging material, as an emulsifier in cosmetics, in the clarification of wine and juices, and in supplements and more. Primex is an Icelandic marine biotech company, and they are our collaborators in this project. And they are a global leader in producing high-quality chitosan from shrimp shell waste. They have several products on the market. Chitoclear is the chitosan that they produce, and they also sell chitosan products such as liposan, which is a dietary supplement that helps with cholesterol and weight loss. Chitocare Beauty is their cosmetic line. Chitocare Medical is their medical device line, mostly focusing on wound treatment, and SeaClear is a water treatment for swimming pools and tubs. And in 2012, they received the Icelandic Innovation Award. So Primex has the advantage of producing high quality chitosan, and they do this under the food safety standard ISO 22000, and they have several other certifications. A couple examples are certified halal product and certified natural product. However, today, the main producer of chitosan is Asia. So while Iceland has the advantage of high quality chitosan, it is important to be innovative and make new chitosan derivatives to stay in the market. So as it has been mentioned before, the main drawback of chitosan is its poor aqueous solubility. So in order to solve this, some chemical modifications are needed. One interesting chitosan derivative is the N,N,N-3-methyl-chitosan, or TMC, which compared to unmodified chitosan has an improved solubility and enhanced antibacterial activity as well, and it is aimed to be used in drug delivery applications. There are several published methods available on the synthesis of 3-methyl-chitosan. However, they have some drawbacks. For example, long reaction time, or using excess of reagents, or resulting in undesired products. And in the synthesis of TMC, it is also important to obtain high degree of 3-methylation for these improved properties. There has been a study in our research group that focused on developing a synthetic method for TMC with high degree of 3-methylation. And in this study, up to 85% degree of 3-methylation issue with this is that in order to obtain this 85% degree of 3-methylation, the reaction had to be repeated 4 times. So we can say that this is not truly a one-pot synthesis. A significant excess of reagent had to be used, and the reaction time was really long. Additionally, later the reproducibility was found insufficient. So we need some optimization. This can be done using the design of experiments approach, which is a systematic engineering method that takes into account multiple input variables, which we call factors, designs the experiments with a minimal number of experiments, and then uses statistical analysis to evaluate the responses. And in a full factorial design with 2 factors on 2 levels, a square can be visualized and the design will consist of 4 experiments, whereas with the 3 factors on 2 levels, a cube can be visualized and the design will consist of 8 experiments. So the aim of this project was to optimize the synthesis of 3MC using the design of experiment approach. We want to obtain a high degree of 3-methylation and high yield, method that has the potential for large-scale production, that is cost-efficient, eco-friendly, and safe, and a product that has the potential for medical application. So first, we started with some we were not working with the base, the reagent, the and by the 5th design, we were able to reach 53% degree of 3-methylation in just 1 step. And compared to the first design, this was an 8-fold increase. It also helped us to establish some reaction conditions that we can further explore in the next designs. And when we compare these results with the previously published method, we can see that much higher degree of 3-methylation was obtained in just 1 round of reaction. So next, with the help of a specialized software called ModDep, a fractional factorial design was done as a screening step to identify the main effects of variables. And the equivalence of the methyl iodide reagent, the equivalence of the sodium bicarbonate base, the temperature, and the time were varied. The highest degree of substitution we obtained here was 59%, which is not a significant increase compared to the previous design. However, at this step, this was not the goal. We just wanted to use the software to see what we can learn about the significance of the factors. And one very important finding was that for both responses, for both the yields and the degree of 3-methylation, the time was found to be non-significant. This means that the reaction doesn't need to be carried out for 48 hours. It is sufficient to do it in much less. So as the next step, a full factorial design was carried out and 2 factors were further investigated, the temperature and the addition of base. And throughout these 12 experiments, up to 72% degree of 3-methylation was obtained with the 2 addition of sodium bicarbonate base, and the reproducibility was found great. However, there was still room for improvement. It was because upon addition of the base, we encountered viscosity issues and gas production. This was not ideal to say, because it made the stirring difficult. And if we want to scale the reaction to large scale or an industrial setting, this is not favorable. So far, we have used 2 bases in the synthesis. So we tried sodium hydroxide and we tried sodium bicarbonate. So as the next step, we tried 7 different non-nucleophilic bases to see if they cause increase in viscosity and produce gas in the reaction. What we did was we performed 2 reactions with each of these bases. First, all of the base was added all at once to the reaction. And in the second reaction, the base was added in 2 parts, 6 hours apart from each other. And then we measured the degree of remethylation and checked the viscosity and the gas production. It was found that there was one base, N-diethyl-aniline, which had no effect on the viscosity and no gas production. However, the degree of remethylation was found to be very low, only 6%, and initially tried some screening and optimization steps, but it was agreed on that it would be really difficult to optimize it from such a low volume. But there was another base, DPL, or N-diisopropylethylamine, which gave quite good trimethylation already, namely 42% when the base was added in 2 steps. There was no gas production, and the viscosity increase was not that large. So we further explored experiments with this DPL base, and we tried it in different solvents. We tried it in DMSO, DMF, and the mixture of DMF and water, and obtained degree of remethylation degrees 60, 67, and 42% respectively, which were quite promising, given that, as I mentioned earlier, that in the previous studies, 85% was obtained by repeating the reaction 4 times, and here we were able to get up to 67% in just 1 round of reactions. Another thing that we tested was repeating the trimethylation. So we took the material that gave 42% degree of trimethylation and repeated the reaction, and were able to obtain almost full trimethylation. We measured 99% degree of trimethylation in that reaction. The next thing we did was we tried the reaction in 3 times diluted media in DMF solvent, just to see if we can further eliminate any remaining viscosity issue. And the base addition and the reagent equivalents were the factors that were studied throughout 12 experiments. The highest degree of trimethylation was again 67%, which was the same that we got before as the highest value. So we could conclude that in a diluted media, we can obtain high trimethylation as well. So the first future perspective of the study is that we just received a 4-zone reaction station from Radley's, and we will carry out the next designs in this reaction station, which will allow us a way more precise control over the reaction. For example, precise heating, active cooling, and software control, and it will also allow us to do 24/7 unattended chemistry. So the next steps are to do more designs with the DPL base and the DMF solvent and see the results. And I would like to thank my supervisor, Professor Marmasson, and Dr. Helen Luzon from Primex. And I would like to thank all the funding. And I would also like to thank you for your attention. Thank you.